Check it out now, y'all.
nanoHUB-U online instruction.
Hello and welcome back to our third lecture in week 5 in nanoHUB-U's Introduction to Bioelectricity. In this lecture, we're going to go back to deep brain stimulation as we did in Parkinson's disease, but we're going to be talking about a different sort of circuit. So in the first lecture this week, we talked about Parkinson's and applying electrodes to the basal ganglia circuitry deep inside the cerebral hemispheres and stimulating a structure further down in the subthalamic nucleus to compensate for the death of neurons in the substantia nigra and the dopaminergic projections of those neurons to the basal ganglia. In epilepsy, we talked about a different circuit, which really revolves around the hippocampus. But more so than that, we were even talking about stimulating the vagus nerve and the projections, the afferent projections of the vagus nerve to the brain in the hopes of affecting the balance of excitation and inhibition around the hippocampus. In this lecture, we're going to talk about an entirely different circuit, the circuit that's associated with the reward pathways of the brain. And this will bring us back to the lecture in week 2, in which we talked about the various different types of neurotransmitters and their role in various different addictive behaviors and illicit drugs. So let's begin by looking at the anatomy of the reward pathway. So the reward pathway in the brain, otherwise known as the limbic loop, is associated with a number of different cortical structures. The anterior cingulate, the orbital frontal cortex, and the amygdala, all on the cortex of the brain, have projections down into the brain to the hippocampus, the orbital frontal, anterior cingulate, and temporal cortex, and from there to the ventral striatum. So the ventral striatum includes a particular structure that we're going to come back to again and again today, which is the nucleus accumbens. The nucleus accumbens and the ventral striatum project to the ventral pallidum, the substantia nigra, both parts of it, and from there, or beg your pardon, to the pars reticulata, the substantia nigra, and from there to the mediodorsal nucleus. Again, remember from our first lecture, when we were talking about anatomical structures, that I said that the name of structures that you'll learn in the brain will often give you a clue as to where they are. So, for example, when we talk about the mediodorsal nucleus, we can infer medial, meaning that it's towards the center of the brain, and dorsal, meaning it's towards the back of the brain. So it'll be towards the center and back of the central nervous system. These projections are each excitatory or inhibitory. So the projections from the cortex are excitatory to the ventral striatum. The ventral striatum is inhibitory towards the ventral pallidum and the substantia nigra, which is in turn inhibitory to the mediodorsal nucleus, which is then excitatory to the cortex. And so you have a loop. And there's a particular balance in this loop, and that balance is key to associating a reward with the expectation of the reward, and then with the pleasurable feelings that arise from receiving a reward that is expected. And both parts of that are equally important. We'll come back to that. It's not just receiving a reward, for example, eating, but it's receiving that reward and coupling that reception with the expectation. So it's not just filling your stomach, but it's filling the stomach along with the act of eating, which tells your body to expect to become full. So those two things have to be coupled in a healthy brain. And here again, we have a coronal slice of the brain, as we saw in Parkinson's disease. And in that coronal slice, you can see the caudate and putamen, which are part of the basal ganglia. They were also involved in motor control. And you see the nucleus accumbens of the ventral striatum and its role in the reward pathway with the projections, the dopaminergic projections from the substantia nigra, which to the nucleus accumbens are inhibitory. So let's look at your brain on drugs. Most of us, when we were kids, got to see commercials of what happens to your brain on drugs, and it's not really a fried egg. If you're old enough to remember those commercials, it's a little bit more like what's shown in this slide. So you have neurons from the ventral tegmental area, and neurons from the VTA, which is right next to the substantia nigra in the medulla. Neurons from the VTA project to neurons of the nucleus accumbens, and they project and release dopamine, neurotransmitter dopamine. These two sets of neurons both receive glutamatergic excitatory inputs from the cortex of the brain. And so you have excitatory descending projections from the brain, and you have a dopamine release between the VTA, the ventral tegmental area, ventral meaning towards the front, tegmental meaning towards the top area, and the nucleus accumbens in the striatum. Furthermore, in the ventral tegmental area, you have interneurons, which act by releasing GABA on the projecting neurons out of the VTA, and GABA, if you recall, is the primary inhibitory neurotransmitter of the brain. And then what we can see is, in these yellow boxes, all of the different, or many of the different illegal drugs, narcotics, that affect this very simple circuit, and how they affect the reward mechanism. And the reason this is important is because affecting the reward mechanism can do one of two things. It can either give you an unearned reward, make you feel good for no reason, or it can take away an earned reward, make you feel bad even though you should feel good. That second category, those agents, I'm sure they exist, but why would you take them? So we don't worry about those. We worry about the first category, the ones that make you feel good even when you haven't done anything that warrants feeling good. And opiates, for example, we'll go through some of these. Opiates act in a number of different areas. Opiates act on the ventral tegmental area interneurons that project inhibitory projections onto the projecting neurons. They also act on the dendrites of the nucleus accumbens. Alcohol acts at the synapse of the interneurons and the projecting neurons within the ventral tegmental area. Nicotine, if you recall, there was a whole category of ion channels of receptors for ion channels that we talked about in week 2, the nicotinic receptors. Well, nicotine acts on the nicotinic receptors in the dendrites of both the dopaminergic projecting neurons in the VTA and the nucleus accumbens neurons that receive the dopamine from them. So it affects the glutamatergic inputs that descend from the cortex. Cannabinoids work on the receptors in the dendrites of the nucleus accumbens, and then cocaine, which is one of the better examples of the mechanisms of addiction, acts, if you'll recall, as a dopamine reuptake inhibitor. And dopamine reuptake inhibitors are fantastic in the globus pallidus because they help with Parkinson's disease, as L-DOPA does. But they're not fantastic everywhere, and they're not fantastic all the time, and they have this undesirable effect of leading to an excess amount of dopamine excitation at the synapse between the VTA projections and the postsynaptic neurons, the nucleus accumbens, by increasing the duration for which a given release of dopamine is present in the synapse. And so what happens when we do that? What happens is that you get simultaneously, when you repeatedly stimulate, overstimulate, by taking an illicit drug, what happens is that you get a desensitization to the stimulus. So you have both an ongoing response and you have a phasic response. The phasic response is the response given to a particular actual stimulus. And that'll be sharpened. And you have the ongoing response, which is your baseline level, and that'll be lowered dramatically. And so what happens is that when you take a drug, you increase that, let's say you take cocaine. You take cocaine, you increase the duration during which dopamine is present in the synaptic cleft between the VTA neurons and the nucleus accumbens neurons. And that leads to an excess of excitation there. So your body is receiving a reward, but you haven't done anything to get a reward. So whereas this reward circuit is very, very important to drive behavior, and it acts to promote activities like eating, for example, is pleasurable because of this reward circuit. Sex is pleasurable because of this reward circuit. So those are specific activities which are coupled to a reward. So there's an expectation of the reward, and there's the activity which delivers the reward, and those are tightly coupled. When you take an illegal narcotic, there is no expectation of a reward because you're not eating and you're not having sex. You're not doing a natural behavior with which you would expect to have a couple of reward. You're just consuming a chemical agent. And so your body receives a reward without the expectation of the reward, and that throws the entire reward circuit out of balance. And so what we need to do is try to come up with ways to restore the balance in that reward circuit. One way is by just stopping whatever chemical agents you're taking. But there's a problem with that. First of all, there's a withdrawal, and the withdrawal comes about because the body compensates for the excess stimulation by decreasing the ongoing amounts of dopamine and glutamate. And so when you stop taking your drugs, your body takes some time to recalibrate and begin producing at the original levels. And so you have, to some extent, you have this delayed effect in which you have to wait for your body to compensate for the absence of that drug. And that's going to feel subjectively exactly the opposite of how the original high felt. And you have to get through that, and that's what we call withdrawal. But in a secondary sense, you have this concern that you are to some extent rewiring the projections in the brain. And so you have plasticity in the brain, and you're changing the wiring of the brain in some cases permanently. And so, or over a much longer period of time than the initial withdrawal. So even though you cease to take a drug, over a period of time, you will go through that initial withdrawal, but it takes much longer than that, and possibly an indefinite amount of time to rewire the brain back to its original state, which is why drug addicts will always tell you that the first high is always the best high, and that when they go clean and then they go back, there's a danger of overdosing because the brain is no longer used to that acute dose because it's recalibrated. But even without the overdose, the high will not be the same as it was the first time. So they progressively become less and less sensitized, and they never quite experience pleasure, let's say in the case of heroin addicts, the same way that they did before beginning to consume heroin. So there are long-term plastic effects as well. In this particular lecture, I want to talk about one example of that, and that is the GABA agonist response of neurons in the nucleus accumbens of alcoholic subjects. So we have alcoholic animals, just like we have alcoholic people, and those are animals that are predisposed to the consumption of alcohol. And there's a particular strain of rats called P rats, and those rats, if you give them the opportunity to consume alcohol, will do so. And in fact, if you take a pup and you put alcohol in their chamber and you put them in the same chamber as their mother, they will preferentially select alcohol to their mother's milk. And they'll drink until they pass out, and then as soon as they're able to walk again, they'll go back and drink some more. So what can we do to change that sort of behavior? 12 steps is not going to help an animal, and so we begin to look at purely interventional engineering approaches. One is to deliver a drug. And so you can deliver a drug that's a GABA agonist. So agonist increases the response to GABA. GABA is inhibitory. So you're increasing the inhibition of the neurons in the nucleus accumbens. And in that way, you're countering the effect of the addiction. And what happens is, in a particular animal, if you have some baseline level of alcohol consumption, over a series of injections, you map the level of voluntary alcohol consumption in the animal. And you can see that the level of voluntary alcohol consumption goes down dramatically, half as much alcohol as they drank before, while you're providing the injections of this drug. And then when you stop providing the injections, it shoots right back up to what it was before. In fact, higher than it was before. And this corresponds with observational behavior, or observations of behavior in addicts. If you administer a placebo, shown here with the black dots, you get no change over the same time period in the level of alcohol consumption. And the asterisks here are to denote that we have a statistically significant difference between the animals on placebo and the animals receiving the GABA agonist. What we want to do is move away from drugs, because drugs are very difficult to inject focally. That requires a focal cannula to deliver the drug. It requires a drug pump, and it requires a reservoir of drug, all of which are very complicated to do in the brain, and especially with our fear of infection of the brain. So to avoid infection of the brain, we take drugs orally, which means that you're going to have a GABA agonist response everywhere in your body, and that's not what you want. You want it in the nucleus accumbens. So we're back to deep brain stimulation. And it turns out that in deep brain stimulation, if you apply an electrical current of 100 microamps to an alcoholic animal, the consumption of alcohol will decrease, but it'll fluctuate, and it won't quite give you the results that you saw in the GABA agonist animal. But if you increase the dose, increase the amount of current, then you get a much more dramatic reduction, almost to a third of the original level of alcohol consumption in the animal. So this is an even better response than we had, and it's statistically significant, an even better response than we had in the original animal, and when we stop the electrical stimulation, the animal immediately goes back to drinking at pre-therapy levels. And all of this work that I'm describing here is the work of a colleague of ours at LSU, Dr. Jessica Wilden, along with a colleague in Indianapolis, Dr. Zach Rod, and a MD PhD student here at Purdue, Kurt Ching. And Kurt's work is what I'll be talking about now. So what Kurt wanted to do is, having looked at this DBS stimulation and the fact that you can reduce addictive behavior without any kind of therapy beyond an electrical stimulus directly into the reward circuit to alter the function of that circuit, he then wanted to say, well, okay, but when we stimulate with these square waves, right? Because that's what everybody does. But is that the best way to stimulate? And so he started to ask if we could make these square pulses out of smaller little pulses, which he calls pulsons, and the square pulses would be composed of the pulsons, and that would achieve two things. First of all, hopefully, it would achieve an improved physiological response. And second of all, it would do so with less current. So if you integrate the area under the curve, you'll find that you have less current being delivered for the pulsons than you do for the square pulse. So you have a smaller dose, which means that you're going to have less desensitization of the brain. You're going to have a less electrodeposition of the electrode material into the brain when you stimulate, even though it's biphasic. You're never perfectly biphasic. And most importantly, perhaps, you're going to have less power consumption of the medical device, which means that your battery life is going to be much longer, which means that your battery can either be much smaller, leading to a less invasive device, or your battery perhaps won't need to be replaced every few years as it does now. And that means surgery avoided. So this approach leads to a series of waveform parameters that we can discuss. One is the amplitude of the pulse. One is the width of the pulse, PW. One is the amplitude of a pulson in a burst. And then the width of the pulson in the burst, AMX and PWX respectively. And then there's the interpulse interval, the spacing between pulsons. And there's the number of pulsons in a particular pulse. There's the effective pulse width, which is the corresponding width to the original pulse. And then there's the burst duty cycle. So each of these can be calculated, and we can look at a continuous pulse versus a continuous pulse chopped up into 2 pulsons, each with a 20% duty cycle, or 5 pulsons with an 80% overall duty cycle, or freeform, where you have little pulsons and big pulsons and all sorts of combinations in between. And if you look at all of these potential waveforms, you can begin to record the response of a volume of tissue to that stimulation. And so you can begin to say, all right, we know that 200 microamps of current works better than 100 microamps of current, but why is that? And is it necessary to go from 100 microamps to 200 microamps, or can we achieve the same effect by playing with that waveform? And so we begin to look at the A, B, and C fibers in a vagus nerve being stimulated with pulsons, and you can look at the peak values and track those. You can look at the width of all of the A fibers firing together. So if they all fired at exactly the same time, you would have a very small width. And if they fire over a period of time that's distributed, you would have a larger width here. And then you could look at spacings between A and B and C fibers. And then, having that information, we can begin to calculate and plot the differential fiber response. So you can look at A fibers at the top, or you can look at C fibers on the bottom, where on the left-hand side, we're looking at measured A fiber and C fiber response in microvolts with different colors for different stimulus waveforms. So the stimulus waveforms are color-coded at the bottom, and what you find is that you have the blue waveform, which is the continuous waveform, has some particular response. And then the other ones, which consist of pulsons, have different responses. And in fact, if you look at the green waveform, which consists of these 2 pulsons with a 20% duty cycle, the green wave shows you that for a given amount of electrical stimulation, you get a much higher level of activation than you would with a continuous pulse. So we do, in fact, see better efficacy with less current rather than better efficacy with more current, as we did in the earlier DBS example where we went from 100 microamps to 200 microamps. So this is a very important finding, and it works for A fibers, it works for C fibers, and we can calculate it also as a percent of the maximal activation. So rather than absolute values, we can look at the maximal activation of the continuous pulse and say, okay, that's maximal activation. And then when we switch to pulsons, we find out that in fact, it's not maximal activation. We can get more than that using pulsons. So you can get more at lower currents and you can get more overall. So if you're just looking at the 50% maximal activation and looking across different waveform types, you can map out what the variation is, and you find that some of these, with the asterisks, are statistically significant, and those show, particularly in the case of the 20% duty cycle, that you have, again, much lower current needed to deliver the same amount of activation. Q50, incidentally, Q is charge required for 50% maximal activation, and the A is of A fibers. So you have Q50A is the charge required for 50% maximal activation of A fibers. Q50C is the same thing for C fibers. So we can reduce this dosing and we can increase the selectivity. That's the take-home message. Using pulsons rather than continuous time pulses, we can reduce the total amount of dosing that is required to achieve the therapeutic effect, and we can increase the selectivity of the fibers that we're interested in to preferentially select some fibers over other fibers to achieve that same therapeutic effect. So whether you're doing deep brain stimulation in Parkinson's disease, in the substantia nigra, in epilepsy, in the hippocampus, in alcoholism, in the nucleus accumbens, wherever it is that you're doing deep brain stimulation, you can use this approach to get more effect with a smaller dose over a longer period of time. And that's where we'll end for today. In the next lecture, we're going to move out of the brain and into the peripheral nervous system and talk about prosthetic arm control. And I'll see you then.